Students, hi everyone, how are you all? I hope you all are doing fine. So in the previous class, we were studying about the properties of genetic material. We also studied the different experiment that took place in search of the genetic material and what are the different experiment conducted by different scientists to find out the genetic material. Right, so we recalled all these experiments also, and we by these experiments and we also studied the properties of genetic material. So we'll recall these properties and we'll move further. So what are the properties of genetic material? First property is the ability to generate its replica, that is replication. Okay, so the process of replication you will study in your further classes. Okay, so in detail you will study, but you just have to know now what is replication. So replica is nothing but Xerox copy. Okay, so you just have to think in way that it is just like a Xerox copy. So if it is a parent DNA, okay, so if it has to transfer its character, okay, to the next generation, that means it has to produce its copy, right? So just think in such a way that this is a parent DNA, right? So ಇದು ಒಂದು ಒಬ್ಬರ parent DNA ಐತಿ. Okay, so a parent DNA, it has to transfer it to their offsprings ಅಂದ್ರೇನು, to the next generation, right? So ಆ next generation ಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗಬೇಕಂದ್ರೆ DNA ಗೆ ಯಾವ ಪ್ರಾಪರ್ಟಿ ಇರಬೇಕು? The DNA should have the property to undergo replication. That means to produce the replica means its copy. Okay, so replication is nothing but the process in which a parent DNA will produce its copies. Okay, so there are two strands of the DNA, right? So these two strands will unwind. That means unwind in the sense they will get separated, and then these strands will synthesize and they will produce two separate daughter strands. So this process we call it as replication. And we also know that the DNA is containing the genes, okay, the genes which will be having the information. So DNA ಗೆ ಏನು ಇರ್ತೈತ್ರಿ? DNA ಅಂತಂದ್ರೆ ಅದು ಏನಿರ್ತೈತಿ? ಜೀನ್ಸ್ ಇರ್ತಾವ DNA ಒಳಗೆ. ಈ ಜೀನ್ಸ್ ಇದ್ ಕೆಲಸ ಏನು? The genes which will have the information to code for proteins. No, so ನಮ್ಮ ಬಾಡಿ ಒಳಗೆ ಯಾವುದೋ ಪ್ರೋಟೀನ್ ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗಬೇಕಂದ್ರು, ಯಾವುದೋ ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್ ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗಬೇಕಂದ್ರು ಜೀನ್ಸ್ ಬೇಕು, ರೈಟ್? ಸೊ ಇವನ್ ಪ್ರೋಟೀನ್ ಸಿಂಥಸಿಸ್ ಆಗಬೇಕಂದ್ರು ಜೀನ್ಸ್ ಬೇಕು. ಆ ಎಲ್ಲ ಇನ್ಫಾರ್ಮೇಷನ್ ಎದ್ರೊಳಗಿರ್ತೈತಿ? ಈ DNA ಒಳಗಿರ್ತೈತಿ. ಸೊ ರೆಪ್ಲಿಕೇಶನ್ ಇಸ್ ವೆರಿ necessity process. So ಇದು ಆಗತೈತಿ in the sense the process of replication, it will help in producing the daughter strands, okay, the copies of the DNA. Okay, so that will transfer the genes, right? That means whatever information the DNA will have, no, that information will be transferred. Okay, so if it is not transferring properly, if there is some mutation, then we have seen there will be many disorders, genetic disorders, be due to the mutation in this genes, right? So this is very important. So first property is what then? So the first property of a genetic material is DNA. So you have studied there are many molecules, right? DNA, RNA, proteins, biomolecules, like there are many other molecules, carbohydrates, right? But they are not the proteins and carbohydrates, they are not called as genetic materials, but only DNA is called as genetic material is because of these all the properties. So we studied first property, which is it should have the replication process. Okay, so ability to replicate, that means it should have to produce its copies. Okay, so how it is done completely molecular level you will study in detail, okay, in the further classes. So first the basic thing you should know that the ability to generate its replica is called replication. That ability a genetic material should have. So next property what it is chemical and structural stability. So the DNA should be, or the genetic material should be, its chemical structure or the structure should be stable. Okay, so it should have this chemical and structural stability. Okay, so next, provide the mutations needed for the evolution. Okay, so provide the mutation needed for the evolution in the sense what? So as we have studied, like before when we were studying the evolution and recombination topics and all, we studied that the mutations, right? So when we were studying about the mutation topic, no, so when we were studying about these topics, we studied that mutation and recombination will lead to the new generation, new kind of species. Okay, so ಈಗ same genes, every time same genes, same characters next generation ಗೆಲ್ಲ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗ್ತಾ ಇದ್ರೆ ಹೊಸ ಸ್ಪೀಶೀಸ್ ಹೊಸದೇನೋ ಯಾವತ್ತೂ ಹುಟ್ಕೊಳಕ್ ಚಾನ್ಸ್ ಇಲ್ಲ, ಹೌದಲ್ಲ? ಸೊ ಫಾರ್ ದಿ ನ್ಯೂ ಜನರೇಷನ್ ಆರ್ ನ್ಯೂ ಸ್ಪೀಶೀಸ್, ಸೊ ಎವಲ್ಯೂಷನ್ ಆಗಬೇಕು. ಸೊ ಇಫ್ ಯು ಕನ್ಸಿಡರ್ the evolution of human beings ಏನಿದ್ರೆ, so if you consider different eras, right? So what you can observe is the human being, he has evolved so much. So now we have the brain which is high capacity, which is having high capacity brain compared to any other organism on this earth, right? We are most developed organisms. But why we are most developed organism? Because our DNA, it provided the mutations which was necessary for the evolution. So we evolved fastly and we are now the most developed organisms. So if these mutations are not always will not always cause the diseases. Okay, so mutations will also lead lead for the adaptation. For example, if there is some mutation in the gene, okay, so ಆ ಜೀನ್ಗೆ ಕಂಪ್ಲೀಟ್ ಆಗಿ ಮ್ಯೂಟೇಷನ್, ಈಗ ಒಂದು ಜನರೇಷನ್ಗೆ ಮ್ಯೂಟೇಷನ್ ಇದೆ ಆ್ಯಂಡ್ ಯಾವುದೋ ಒಂದು ಡಿಸೀಸ್ ಇರ್ಬೋದು ಅಥವಾ ಯಾವುದೋ ಒಂದು ಹೊಸ ಕ್ಯಾರೆಕ್ಟರ್ ಆ ಮ್ಯೂಟೇಷನ್ ಇಂದ ಆಗ್ತಾ ಇದೆ ಅಂತ ತಿಳ್ಕೊರಿ. ಸೊ ಬರ್ತಾ ಅದೇ ಮ್ಯೂಟೇಷನ್ ಆಗ್ತಾ ಇದ್ರೆ ಏನಾಗ್ತೈತಿ, it will get evolution. Okay, ನಾವು ಅದಕ್ಕೆ ಅಡಾಪ್ಟ್ ಆಗಿಬಿಡ್ತೇವೆ. ಸೊ ಅಡಾಪ್ಟ್ ಆಗ್ತಾ ಅದು ಒಂದು ನ್ಯೂ ಕ್ಯಾರೆಕ್ಟರ್ ಆಗಿಬಿಡ್ತೈತಿ, ರೈಟ್? ಸೊ ಒನ್ ಸಿಂಪಲ್ ಎಕ್ಸಾಂಪಲ್ ಐ ವಿಲ್ ಗಿವ್ ಯು. ಸೊ ಯಾವ ಎನಿಮಲ್ಸ್, ಓಕೆ, ಹ್ಯೂಮನ್ ಬೀಯಿಂಗ್ಸಿಗೆ ಮುಂಚೆ ಏನಿತ್ತು, ಬಾಲ ಇತ್ತು, ರೈಟ್? ಮಂಗನಿಂದ ಮಾನವ ಅಂತೀವಿ. ಸೊ ಹ್ಯೂಮನ್ ಬೀಯಿಂಗ್ಸ್ ವೇರ್ ಹ್ಯಾವಿಂಗ್ ದ ಮ್ಯಾನ್ ವೇರ್ he had a tail. Okay, but by the evolution process, andre, so this tail was not used. So you will also study use and disuse theory. This tail was not used by man. Okay, so as the time passed, the tail, it became shorter and shorter, and now do we have tail? No, we don't have tail because of the evolution, because this tail was of no use, right? So it was of no use, so it got extincted. The tail is not seen in the human beings, right, in the man now. So that means the mutations and changes, so they are needed for the evolution. Okay, so the mutations also will lead to different ಕ್ಯಾರೆಕ್ಟರ್ಸ್. ಸೊ ಯಾವ ಥರ ಒಂದು ಆರ್ಗನ್ನ ನಾವು ಜಾಸ್ತಿ ಯೂಸ್ ಮಾಡಿಲ್ಲ ಅಂತಂದ್ರೆ ಆ ಆರ್ಗನ್ನಿಂದ ಏನು ಯೂಸ್ ಇಲ್ಲ ಅಂತಂದ್ರೆ ಏನಾಗತ್ತೈತಿ, ಸೊ ದ ಆರ್ಗನ್ ಈಸ್ ಎವಲ್ಯೂಷನ್ ಆಗ್ತಾ ಡೇ ಬೈ ಡೇ ಏನಾಗತ್ತೈತಿ, ಆ ಆರ್ಗನ್ ಡಿಸಪಿಯರ್ ಆಯ್ತು. ಓಕೆ, ಲೈಕ್ ದ್ಯಾಟ್ ಓನ್ಲಿ ಮ್ಯೂಟೇಷನ್ ಅಂದ್ರೇನು, ನ್ಯೂ ಚೇಂಜಸ್ DNA ಒಳಗೆ ತರ್ತಾ ಇದ್ರೆ, ಸೊ ಸಮ್ ಟೈಮ್ಸ್ ಆರ್ ಮೋಸ್ಟ್ ಆಫ್ ದ ಟೈಮ್ಸ್ ಅದು ಹಾರ್ಮ್ಫುಲ್ಲು ಇರ್ಬೋದು, ಬಟ್ ಸಮ್ ಟೈಮ್ಸ್ ಅದು ಯಾವುದೋ ಒಂದು ನ್ಯೂ ಕ್ಯಾರೆಕ್ಟರ್ ಫಾರ್ಮೇಷನ್ ಆಗಕ್ಕೆ ಟ್ರೈ ಮಾಡ್ತೈತಿ. ಆ್ಯಂಡ್ ವೆನ್ ದ ನ್ಯೂ ಕ್ಯಾರೆಕ್ಟರ್ ಇಸ್ ಫಾರ್ಮ್ನು, ಸೊ ವೆನ್ ಇಟ್ ಈಸ್ ಗೆಟ್ಟಿಂಗ್ ಅಡಾಪ್ಟೆಡ್, ಸೊ ವೆನ್ ಇಟ್ ಈಸ್ ಗುಡ್ ಆ್ಯಂಡ್ ವೆನ್ ಇಟ್ ಈಸ್ ಗೆಟ್ಟಿಂಗ್ ಅಡಾಪ್ಟೆಡ್, ಸೊ ಇಟ್ ವಿಲ್ ಕ್ರಿಯೇಟ್ ಅ ನ್ಯೂ ಸ್ಪೀಶೀಸ್. ಸೊ ದ DNA, it should also provide, or the genetic material should also provide the mutations which are necessary for the evolution. Correct agi evolution agi new species barlikke adu help madbe. Okay, so next, ability to express as Mendelian characters. So the genetic material should also have the ability to express the Mendelian characters. Okay, so it should also express as the Mendelian characters. We have seen all the genes and everything, so many of them they inherit in the Mendelian characters, so which is very very important. Okay, so that means we can get the true species, okay, the pure breeds, okay, we can get the the heterozygous breeds, right? So they should have the ability to express as a Mendelian character. So very very important concept you should remember. So first thing, for an element or an material to be called as genetic material is it should have the ability to generate its replica. It should be structurally and chemically stable, okay, and then it should have structural and chemical stability, and it should provide the mutation needed for the evolution and ability to express as the Mendelian characters. Okay, so this is very very important. So here we have just shown what is actually happening. So the DNA, it will undergo transcription, okay, and then it will produce RNA, and then it undergoes translation to produce amino acid chain, which will undergo different folding to form a protein. Okay, so this process we will study in the next concept. So here what you have to just study is these four different properties of the genetic material. So in the further classes I will explain you, and you will get to know in detail what is this transcription, translation, how the proteins are formed by the DNA. Okay, so reasons for stability or less reactivity of DNA, and reason for mutability or high reactivity of RNA. So here we have to just study why the DNA is more stable than that of the RNA. So which is more stable? DNA is more stable than the RNA. So which is more reactive? So if it is not more stable, okay, so it undergoes RNA, it undergoes mutation compared to DNA. So DNA also mutation occurs, but compared to RNA, DNA is it is more stable. That means it is less reactive. Okay, so the DNA is stable in the sense it will not react more. Okay, so the stability of the DNA, which is less reactive, and RNA, which because it is more reactive, it is prone to mutation. So reason for mutability or high reactivity of RNA we call it. Okay, so first thing, why the DNA is more stable than RNA? First thing, we got to know that RNA is more active than compared to DNA. DNA is less active, right? And DNA is double stranded compared to RNA. So double stranded is nothing but it is having its partner, right? So but RNA is single stranded. Okay, so it is not having its partner, so it is not stable. Okay, in that way you can remember. So it is double stranded and this is single stranded, and then presence of thymine here and presence of uracil, so very very important, and then absence of T2-OH in the sugar, whereas 2-OH in the presence of 2-OH in the sugar. So as we have seen, deoxyribonucleic acid we call it because the DNA, which is deoxyribonucleic acid, it is not having this 2-OH in the sugar molecule, but the RNA is having extra 2-OH group in the sugar molecule, that you can see here. So deoxy in the sense what? The oxy meaning oxygen, right? So here you can see deoxyribose, that means the ribose sugar which is deprived of oxygen, so only H is present. Okay, so but here in case of RNA, it is just ribose nucleic acid. Okay, so ribose nucleic acid, so OH group is present here. Okay, so here only H group, H is present, that is deoxy, that means oxygen is absent, whereas here OH group is present. Okay, so that is leading for the stability of the DNA. Okay, so this in detail structure you will study further. Okay, thus RNA is unstable, so RNA viruses, okay, for example QB bacteriophage, tobacco mosaic virus, mutate and evolve faster. So since RNA which is almost it is present in the viruses like bacteriophage, QB bacteriophage, okay, and then tobacco mosaic virus. Okay, so these, because they are having RNA as the genetic material, so RNA is unstable, so in such viruses they will mutate and evolve faster. So they will mutate and they will evolve faster, they will be having new species, new generation faster. Okay, so even this coronavirus, as you can see, even it is going in mutation. Okay, so the mutation which are causing different stains, right? So different stages we are going through because of these mutations in the corona, right? So that also you can find out whether corona is having DNA or RNA. Okay, so what it is, so it is also a virus, right? So what it is doing, right? So you have to watch if you are interested in biology, no? So don't expect everything from a teacher, right? So you just go and search what is this. So we are studying the same thing, no? We are studying the mutation, we are studying about the genes, so you have to even watch what is going in day-to-day life, right? So day-to-day life, now they are talking about third wave. So what do you mean by this third wave then? Okay, so what do you mean by third wave? What is happening actually in third wave? So why the new coronavirus now, again new strain they are telling? Okay, so it is very dangerous, like that they are telling. So what is the reason? So what is actually happening to the coronavirus? So everything, if you take this subject, no, if you study just as a theory and biology, it is subject, we have to write the exam, then you will just be in the box. Okay, so whatever is there, that only you will study, but just go out of the box, relate it to whatever is happening around you. Okay, so you can relate biology specially to your environment and whatever is happening around you, so that you will get to know. Okay, so I may forget later, but I want you to do a small research on this. So you don't have to go to hospitals and do research what is happening, or go to doctors. You can sit at home, you have the wonderful device with you, that is your phone, that is your mobile. Okay, you can search, you can Google any information and you can note down. Okay, so you can learn. So mutations. Okay, what we were studying, the properties of genetic material. So RNA is unstable, and that is why the viruses which are having RNA, QB bacteriophage, tobacco mosaic virus, they will undergo mutation and they will evolve faster. Okay, so and DNA are complementary. Okay, so DNA strands are complementary. On heating they separate, in appropriate condition they come together. So in Griffith's experiment, some properties of DNA of the heat killed bacteria did not destroy, so it indicates the stability of the DNA. So you can see in the Griffith experiment we saw that heat killed the S stain. Okay, so S stain, so that bacterial cells he heat killed and he transferred and he mixed with the R stain. Okay, but still the DNA did not destroy from the S stain, it still get transferred into the R stain. So it shows that DNA is stability. It shows its stability, right? So DNA, if it is double stranded, so on heating, if you heat the DNA, so then it will unwind in the sense it will open, the strands will open, the separates, okay. But on cooling, the strands will again rewind, okay. So again you will study in the next classes, okay. So complementary in the sense, why they are called as complementary? If there is the codes which are always complementary, if it is A, then it should have to complement with T. So G, it will complement with C, and it will have the complementary stand like that. All the codes A, T, G, C, okay, they'll be like this, and they will complement with each other. So stands are completely, their codes are so much complementary, okay. So for the storage of genetic information, DNA is better due to its stability, but for the transmission of the genetic material information, RNA is better. So RNA can directly code for the protein synthesis, hence can easily express the characters. DNA is dependent on the RNA for the protein synthesis. So this is very, very important that you should remember. So for the storage of the genetic information, DNA is better because DNA is, as we said, it is more stable. So you can store it as as a genetic information. So storage of genetic information in our body, DNA is more suitable because it is more stable, okay. But for the transmission of the genetic information, RNA is better, okay. So because you will study in transcription and translation process why RNA is better, okay. RNA can directly code for the protein synthesis and can easily express the characters. So DNA, it will undergo transcription to produce RNA, and RNA, it will undergo translation to produce protein. So in this slide, it is not that easy to just understand this process by just this slide. So you just have to remember the DNA, which is more stable as a information to store the information, but DNA is depending on this RNA for the transmission of the genetic information. So the DNA, okay, RNA, it is formed by the DNA itself, okay. You will study in transcription process. So RNA is formed by the DNA, and this RNA, it will help in the protein synthesis because it is, it will code for the proteins, okay. The transmission of information is better by the RNA. So this just you have to remember the DNA. Transcription, it will produce RNA, and then translation, it will produce protein. So how it is done, we will study very depth and very in detail process in the further slides. What is transcription and translation, okay. So just you have to remember DNA is depending on the RNA for the protein synthesis, and not just the viruses will have the RNA. So as the genetic material, they'll have the RNA, okay. But in case of human beings or in higher organisms, the genetic material will be what? DNA. That means from parents to the offsprings, the DNA will only get transferred, okay. So parents to the son or daughter, okay, for the next generation. But for the synthesis of the protein, the RNA is required. So even the human beings or any higher animal, they need RNA for the protein synthesis, okay. So we will study how and how it will be done, okay. So RNA was the first genetic material. So they say that RNA was the first genetic material, okay, but it acts as a genetic material and also as a catalyst. So what is actually a catalyst? Catalyst is nothing but the something that will act as an enzyme, or like enzyme like, Okay, so it is mediator. Okay, it will help in, it will not completely evolve itself, but it will help in some process that we call it as catalyst. Okay, so genetic material, it acts as genetic material and catalyst. Okay, so essential life processes like metabolism, translation, splicing, etc. are evolved around RNA. So splicing you will learn further for in the protein, after the protein synthesis. So you have these all things, whatever you are watching now in this slide, you will study for completely in depth in the further slides. So RNA, you just have to remember in this slide is nothing, is just that RNA is the first genetic material found. Okay, and then it acts as a genetic material as well in some organisms, as well as it acts as a catalyst. Okay, which will help in catalyzing the processes, and also it is essential for metabolism, translation and splicing, that you will learn in the further slides what it actually means. And DNA is evolved from the RNA for stability. That means actually the genetic material was, first genetic material was only RNA. Okay, since RNA is very unstable, okay, it is highly reactive and it is unstable, so to get the stability, RNA it developed as a DNA. So like that, DNA it also evolved from RNA. Okay, so for a stability. Okay, so this you have to remember. So we just saw the slides, the properties of genetic material and why the DNA is less reactive or high stable than that of the RNA. Okay, and then we studied what actually is happening in the protein synthesis, how the protein synthesis takes place. It is from DNA and RNA and then protein. So that process only now we will study. So this we call it as central dogma of molecular biology. So whatever I have taught you now, you will be having many of the confusions. What madam is telling, what is this transcription, what is this translation, what is this splicing. Okay, so what she is telling about, what she is talking about. So this is not just not the topic that can be explained just by a single sentence. Okay, so this we call it as central dogma of molecular biology. So what do we call it as? Central dogma of molecular biology, where the DNA, which is the genetic material, which is having the genetic information, which will code for the RNA, and this RNA which will help in producing the protein molecules. Okay, and then splicing and all, they will come after the protein molecule is produced. So this we call it as central dogma of molecular biology, that we are going to study in detail. And after this explanation, whatever the doubt you have here, no, so what is this transcription, translation, what madam has taught, RNA, DNA is dependent on RNA, what she has taught, everything, every doubt will be cleared in this video. Okay, so here after completing this central dogma of molecular biology, so first is this central dogma of molecular biology. It is proposed by Francis Crick. Okay, where you have heard this name already before, Watson and Crick, right? So Francis Crick, okay, he proposed this central dogma of molecular biology. So it states that the genetic information flows from DNA to RNA, then to protein. So what it says, the DNA, okay, it undergoes replication, and then since the DNA is having the property to replicate, because we have studied, so replication is the property of any genetic material. It should be able to send its or form its replica. Okay, so the DNA ಏನೈತಿ ಪ್ರಾಪರ್ಟಿ ಏನೈತಿ DNA ಇದು ರೆಪ್ಲಿಕೇಶನ್ ಮಾಡಬೇಕಂದ್ರೆ ಅದರದ್ದು ರೆಪ್ಲಿಕಾ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಬೇಕನ್ನೋದು. ಓಕೆ, ಸೊ ವೆನ್ ದ ರೆಪ್ಲಿಕಾ ಇಸ್ ಪ್ರೊಡ್ಯೂಸ್ಡ್ ಆರ್ by the transcription process, the DNA will form the RNA. Okay, so and then from RNA, that by the translation process, it will form the protein. So one by one, transcription and translation we will study. So in some viruses, flow of information is in the reverse direction, that is RNA to DNA. It is called as reverse transcription. Okay, so actually how the genetic information flows, it flows from DNA to RNA and RNA to protein. Okay, so how it flows, it flows from DNA to RNA and then RNA to protein. Okay, and in some viruses, no, it is reverse. Okay, so it is transforming from RNA to DNA. We call it as reverse transcription. Transcription is happening reverse direction. It is happening in RNA to DNA direction. So we call it as reverse transcription. Okay, so till here I am sure you will be having one or the other doubt. So what is this transcription? What is ma'am teaching? What is this reverse transcription? But actual information which will, whatever the doubt you'll be having, it will be cleared from here. Okay, so this concept, after completion of this concept, one we will recall, and they all are interrelated, and we can sum up as a whole and we can learn. Okay, so DNA replication. Then what do you mean by replication? Replication is nothing but copying of the DNA from the parental strand. Okay, parental DNA. Okay, Francis and Crick proposed the semi-conservative model of replication. Okay, so Francis and Crick, Watson and Crick, sorry. Francis Crick it is, not Francis and Crick. Crick's name is Francis Crick. Okay, so Watson and Crick, they proposed the semi-conservative method of DNA. It suggests that the parental DNA strand acts as a template for synthesis of new complementary strand. After replication, each DNA molecule would have one parental and one new strand. And Matthew Meselson and Franklin Stahl 1958 proved semi-conservative model. So replication is what? So don't have to be panic or get scared of whatever about the topic. So be very attentive, and it is very interesting topic. So you have seen that the chromosomes will get transferred from parents to the offsprings. That means what is actually happening in the DNA. Okay, so what is actually happening and how the DNA will get replication, produce its replica, that we will study. Okay, so replication is nothing but copying of the DNA from the parental strand. As you can see in diagram, one, it is parental DNA. So these two strands, they are getting unwind. Okay, and then from these two strands you can see there is one more strand that is getting synthesized here. Okay, red color. So like this the daughter DNA will be synthesized. It is not that easy how I am showing. So it is also involving many molecular and bases. Okay, so you will study one by one further. Okay, so the DNA, the replication is nothing but copying of the DNA from the parental strand. So and that Watson and Crick, they proposed the semi-conservative model. So what is semi-conservative model of replication? That we'll see. Okay, and that suggests that the parental DNA strand acts as a template in the sense it acts as a constant template by which the other strand will synthesize. Okay, so for the synthesis of new complementary strand, after replication the DNA molecule would have one parental strand and one new strand, and you can see here each of the daughter DNA, they will have their parental strand, one parental strand, and they'll have one new strand here. So in each of the daughter DNA you can see there is one parental strand, one new strand which is synthesized. Okay, so and Matthew Meselson and Franklin Stahl, they proved that it is semi-conservative model. Okay, so that we will study what is actually semi-conservative model. So students, I think today we will stop here, because I know you people might be very confused. So you don't have to get confused. It is very interesting and very new topic for you. So you will study how the genetic genes are actually, how they are coded, okay, how the proteins are actually produced from these genes, so how the DNA will help in production of the proteins, okay. So what is this RNA acting here in between? So very interesting and very deep concept, okay. So I think we'll stop here, and in the next class, okay, whatever the doubt you will be having, you yourself will clear it one by one. So that much interesting the topic is. So here just they have given the theoretical view of whatever you are going to learn, but actual process of transcription, translation, protein synthesis we will study. So here in this class you just have to remember the properties of genetic material that we have studied, okay, and what is the difference between DNA and RNA, and what are the complementary strand, how they are produced, okay. So DNA, it will by heating it will unwind, okay, and then the two strands are separated, and by cooling the strands will be rewinded, okay. And then we just studied, just you should have the knowledge just that the DNA will produce RNA by the process of transcription, and then RNA by the process of translation it will produce protein. So how it is done we will study one by one, and then you just have to know that RNA was the first genetic material, and this RNA, it will act as the genetic material as well as a catalyst, okay. And DNA, so first genetic material was RNA, and to synthesize but RNA was unstable, so to make it stable, so complementary strand, everything it got evolved, and then the DNA is evolved from the RNA to get stability, okay. So and then central dogma of molecular biology. So what is central dogma of molecular biology? That the DNA, which is genetic material, it will undergo transcription, it will produce RNA, and then RNA undergoes translation and it will produce proteins. This itself is a central dogma of biology, which is molecular biology, which is proposed by Francis Crick, okay. So this much you have to remember, and everything else in detail, whatever actual processes is there, whatever transcription, translation, we will study replication first of all, and semi-conservative method, everything through the different diagrams and experiments we will study one by one. So don't have to worry, we'll make it more interesting. And remember the homework that I have given. So to see this science is such a way that if you take it like an exposure, okay, so if you take it as an adventure, okay, so then it will be more interesting. So you yourself will start thinking until, so if you are really interested, so until I take next class, you will be always having the doubts in your mind, how it is, how come it is like that, how come it is like this, like that, okay. So it is that interesting, and you just have to put your interest and explore the surroundings and your related topics, okay. So you have to relate it to new topic, and whatever it is there you are observing, so then it will be more interesting. So for you the homework is, you have to collect the information, what is actually happening with the coronavirus, how it is getting mutated, and what is actually this new strain, and whatever new wave, why they are calling, okay. So thank you. We will study the central dogma of molecular biology and DNA replication and all in the next class. So if you have not subscribed to the channel, please subscribe, and you watch the video but you don't hit the like button, so press the like button if you enjoy.